A fascicle is a group of myofibers bundled together. One myofiber contains many myofibrils. Myofibrils are grouped together in a bundle surrounded by a plasma membrane called the sarcolemma. Within the sarcolemma, myofibrils are bundled together with nuclei, mitochondria, and a fluid-filled membranous network called the sarcoplasmic reticulum. The sarcoplasmic reticulum encircles each myofibril and is fed by transverse tubules, or T-tubes. The T-tubes are open to the outside of the sarcolemma. They are filled with interstitial fluid, and they work with the sarcoplasmic reticulum to ensure that an action potential will excite all parts of the muscle fiber at exactly the same time. The basic functional unit of a myofibril is the sarcomere. The Z-disc is a narrow, dense area of protein material that separates one sarcomere from the next. A sarcomere consists of both thick, in the red, and thin, in the brown, filaments. The dark middle part of a sarcomere is called the A-band. The A-band is densely packed with both thick and thin filaments. The H-zone is a narrow region in the center of each A-band. The H-zone contains thick filaments but no thin filaments. The area with the lightest density, containing only thin filaments, is called the I-band, and a Z-disc passes through the center of each I-band.
Myofilament is composed of thick filament and thin filament. Thick filament contains myosin protein, and thin filament contains actin protein, troponin, and tropomyosin. Calcium floods over the thin filaments and binds to troponin and pull the tropomyosin off the active site on actin filament. High energy myosin head leap to bind on the exposed active site on the thin filament, forming a cross bridge. Myosin heads bind to actin, forming cross bridges. Myosin head rotates toward the center of the sarcomere and is called the power stroke. As myosin heads bind to ADP, the crossing bridge are detached from the actin site. Contraction cycle continues if ADP is available and calcium level in the sarcoplasm is high.